Hello everyone, welcome to MACSMIN2025. Now Jeffcott will talk about quantifying geometric variability and atom clashes in protein structures. Please, Will.
Have you got the volume on your laptop or?
No.
Okay, I'll turn it on mine then.
Yes.
So yeah, we can potentially hear if anyone's got questions. Yeah, hello everyone. So continuing with the theme of proteins from Olga's talk, I'll be talking about quantifying geometric variability and atom clashes in protein structures using the protein data bank. So the introduction is quite similar to Olga's really when we talk about the structure of proteins. So we've got these amino acid chains where we're just given like a list of residues. We build up to sort of secondary structures that are like things like alpha helices and beta pleated sheets. And then there's these sort of tertiary and quaternary structures which represent how they actually fold up in three-dimensional space. So we've got the protein data bank that's essentially a global repository which includes all of these 3D coordinates, and the structure is essential basically for fields like drug design, where going back to the lock and key theory that Olga mentioned, the structure of them is essential. The last talk talked about the backbone rigid invariant, which was from our paper. So that was quite abstract, but it was a complete invariant under rigid motion, which was the sort of essential result. In this study, we'll focus on a slightly less abstract invariant, which is more familiar to bioinformaticians, based on bond lengths, bond angles, and torsion angles. And the goal really of this is to sort of quantify deviations within the PDB, highlight some features that should be sort of physically unlikely, such as atom clashes. And essentially just show examples where there's quite a bit of deviance and we might see them as a bit of an outlier. So going back to some of the definitions again that Olga used, we've got the backbone that is essentially the sequence of atoms along the core of a polypeptide chain. So in our analysis, we restrict to the sort of main nitrogen, denoted by N, alpha carbon, denoted by A, and carbonyl carbon, denoted by C atoms. And they've got this corresponding subscript of I, which indicates they're in the ith residue. So you can see from the picture here on the right-hand side, we've got, we start at sort of a carbonyl carbon from the previous residue. Then we go along sort of NAC. These are all from the ith residue, and then onto the next residue. And labeled on these diagrams is both the length. So we use the notation length from the sort of atom on the left to the atom on the right. This one's length Ci-1 to Ni, and so on. And then there's also these bond angles. So the notation here is the sort of three atoms that they're an angle between in sequential order. So we're basically going to, in this talk, explore so-called geometric outliers that are parameters which deviate beyond statistical thresholds. So in this talk, we've defined them as sort of 3 sigma, 5 sigma, and 10 sigma away from the mean across the entire PDB. And we'll also talk about atom clashes as well, which are cases where bond lengths or distance between backbone atoms are less than 1 angstrom, which sort of seems physically impossible due to interatomic forces and the sort of energy it would require to put two atoms that close together. So just further definitions, we've also got these dihedral angles, which in the literature are generally referred to by phi, psi, and omega, respectively. Here we'll use slightly different notation again. So we've got these tau notations. So basically, the dihedral angles are the angles between two half planes that span. So the sort of three previous atoms and then the three next atoms. So in this case, for example, tau and then Ci minus 1, Ni, Ai, then Ni, Ai, Ci. So that's the plane that spans these three on the left and the plane that spans these three on the right. And then we figure out the angle between them. So sort of the notation here is a bit more precise. And again, they're indicated on the graph along with their sort of traditional notation. So you can see this one, which is that one's phi I, psi I, and omega I plus 1. So yeah, there's the omega one is the one across the peptide bond. So our combination of these three lengths, three angles, and three dihedral angles for each residue forms a complete invariant. Now the dimension of this invariant, so we had in the BRI, that the dimension was m times 9, where m was the length of the protein chain, and 9 was represented the fact that we had three coordinates in each three of the basis vectors. Here we've got m times 9 minus 6 values, and this is just because the minus 6 is due to the fact that we don't have angles defined on the end residues, and similar with the dihedral angles, we don't have them near to the ends on either side. And this forms a complete invariant, essentially, which we've discussed in the first two talks, but essentially this is an invariant, but with the additional property that the invariant values are equal if and only if the original molecules are equivalent. So here we choose the equivalence of two proteins being rigid motion as well, as usual. So here's just a snapshot. It's sort of quite similar to Olga's, although it was based on a sort of slightly updated view of the protein data bank. So I think in Olga's analysis, there were 703,000 protein chains after filtering. This used an identical filtering process, but it was just done sort of roughly, I think, roughly a year later or so. So the number of chains that we had to look at had gone up to 753,000. Again, there's just an example on the right here of some sort of fairly common proteins in biology. And as Olga said, they're just sort of artistic representations, but I think they show sort of how complex and varied various proteins can be. Again, there's this sort of feature that to get all this data and is sort of relatively quick and can be done on a modest laptop, which is sort of an advantage of sort of our process, but also, yeah, the PDB and the various APIs and things. So the first thing to focus on is atom clashes. So as we defined previously, it's when sort of two atoms are within an angstrom of each other within the backbone. So these can either occur sort of within the sequential atoms along the backbone. So we've defined these in this table as the number of clashes went within that particular length. So if that length is less than 1 angstrom, we've classified it. Similar with the other two lengths, but we've got this, let me just move that. We've got the third one as well, which is other clashes. So that's clashes where it's not necessarily sequential atoms, it can just be other atoms. So maybe where it's sort of folded back in on itself. Yeah, so we essentially scanned the entire 753,000 proteins in the database. We found sort of 63 examples of this, where they're less than one. And this table essentially shows the 20 most prominent cases. So this is where it's ranked basically in terms of number of clashes from most to least. So we see some quite prominent examples here. It's also worth noting that they're sort of X-ray structures with quite low resolution as well in some cases. So it's not necessarily that you would think that they were poor quality. Yes, yeah, sorry. Yeah, that low means good, sorry, in this case. Yeah, so like less than 2 angstroms is yeah, quite detailed really. So you've got this example at the top that's, you know, it's got 18 atom clashes in it, but it is allegedly a XRD structure with resolution of 1.98 angstroms. So, I mean, one thing to consider is just, like Olga says, when we're training things like AlphaFold, do we really want examples like this within our sort of ground truth data set? Yeah, should sort of future predictions be based on learning from, yeah, examples that we might think would be erroneous? And we can also just look at distribution of the various invariant values. So here we've got sort of just simple histograms for length, various angles, and then the sort of dihedral angles are plotted in sort of circular parametric plots, where sort of red indicates higher concentration. So it would be like at the x-axis, this way is like an angle of 0, and we sort of increase the angle as we go around. So we can see sort of quite sharply peaked histograms. It's also worth noting, sorry, that they're plotted on a log scale. So yeah, these, they probably look broader than they are in reality, these peaks, but we can see that there's sort of plenty of examples of lengths and angles as well, really, that deviate quite a lot from the mean, which, you know, could be some sort of natural geometric variability, but in general, these things are sort of plotted to or refined using like idealized values. So yeah, it can be quite, particularly for this length, CI minus 1 NI, there's quite a lot of a sort of tail of longer bond lengths. Yeah, so there's can be quite a bit of deviation from ideal values.
Sorry, what is ideal?
Well, in this case, we've sort of defined as the mean. I think a lot of structural refinement software generally have some sort of idealized angle for the bond. I think it can vary slightly between different software packages, but I think it's always roughly the same, and generally agrees closely with the kind of means that we've got as well. Yeah, so then this is just in sort of table format as well, some of those distributions, yeah, with concrete figures. So we've omitted the dihedral angles here just because they sort of have multiple modes and generally occur across the entire range of their definition. But yeah, we sort of, we can define outliers really in a number of ways, but the way that we've chosen to do here is to consider the sort of standard deviation of these distributions. So we've got mean, median, that generally is identical roughly to the mean, showing that there's sort of quite symmetric distributions. but yeah, we've got these pretty small standard deviations in general. So we can define ranges based on the mean plus or minus either 3, 5, or 10 times the standard deviation. And then we can make these plots of outliers, basically. So we've got the length and angle outliers here. So basically, we've plotted chains. The count on the y-axis is the number of chains. So we've plotted chains where there's at least one or more outliers up to 10. I can see, yeah, so there's generally quite a lot of the 753,000 that have at least one outlier in either the angles or the lengths. But this is only 3 sigma, so it's quite, it's not the sort of strictest definition of outliers. We can see again, if we increase that to 5 outliers, that a lot of these counts have gone down, so they're more generally sort of in the tens of thousands that have at least one residue with an outlier plus or minus outside plus or minus 5 standard deviations. So there's 179,000 chains that have at least one, and then in this definition there's 22,000 that have at least 10 across the 6 invariants. And then lastly, 10 sigma. So this is quite a strict definition, really. Yeah, so these are sort of angles and lengths that are really deviating from the mean across the entire PDB. We can see in this paradigm there's sort of 21,000 chains that have at least one outlier when all the bond lengths and angles are considered. So again, if we're going on to train some form of neural network based folding predictor or something like that, so something like AlphaFold, we might want to get rid of these from our ground truth data sets that we use for training. So then we can go on and use the data that we've got to essentially visualize the PDB by these geographic style plots. So quite a common one to bioinformaticians is the so-called Ramachandran plot, which essentially plots the distribution of 2 of the 3 dihedral angles. And this is really sort of what determines the shape of the protein backbone in 3 dimensions, because it, yeah, basically determines how it sort of twists as it snakes on. So yeah, this is quite a sort of famous picture, really, where we have these regions of high concentration, and they're sort of generally attributed to like certain secondary structures. So this area of concentration is generally attributed to like alpha helices, I think. So the key thing to note here, so our plot on the left is from our entire filtered subset of 753,000 protein chains. And the key thing that has been pointed out when we've shown this to bioinformaticians in the past has been there seems to be quite a lot of residues in this middle bit that are generally thought to be sort of inaccessible for sort of like stereochemistry reasons, like certain atoms being too close together and things like that. Yeah, so we see quite a lot of concentration in this area when we generally expect it to look more like the plot on the right. So our hypothesis really was maybe it's due to a lot of low resolution structures. So what we did for this plot on the right was we sort of filtered our PDB based data set to just include chains that are from experimental method of X-ray diffraction and have a resolution of less than 2 angstroms. So the chains that are generally regarded as higher quality within the PDB. And from this, we saw, yeah, a lot less in those sort of central regions, which was more as expected. So again, going back to sort of training neural network algorithms, you might want to consider just the resolution of this data really when training them. And we can go even further with this. So the previous example was an example of a pairwise plot with 2 of the 9 invariants. So it was 2 of the dihedral angles. We can do this though for all lengths, angles, and dihedral angles. So we can make 9 pairwise plots. So if we restrict to the sort of length pairwise ones, we can see the distribution of 2 lengths side by side. I mean, in an ideal world, we'd like to do all 3 lengths at once, although it's quite hard to visualize with it being 3D. But yeah, we can see sort of these 2D snapshots. So we can see the sort of high concentration areas, which is red, around the sort of idealized lengths. And then we see sort of, it's relatively symmetric distribution as we go outwards. But we can see sort of plenty of outliers on the periphery here. And it's the same in sort of all 3 plots, really. Similarly, we can do the same for angles, right? So we've got these similar plots where we have high concentration around the most common angles, and we can see sort of residues as we go further out. So there's quite a lot of ones which we might sort of deem to be outliers as we go further out. And then lastly, we've got the dihedral plots. So we've got this Ramachandran one that we already showed in the middle. And there's 2 other ones, right, with the sort of 2 other possible combinations. So we can see from these as well that we've got this feature of the omega angle, where it generally goes into sort of 2 expected modes at 0 degrees. And then we've got this discontinuity, which Olga mentioned, at plus or minus 180. So that's basically the same thing. We've just sort of split this peak into 2. So we can see, as expected, really, we've got a lot more of the plus or minus 180 case, which is generally referred to as the trans case of this dihedral angle. And then we've got sort of some concentration, but it's much less common at 0. But then we've got plenty of things in between as well, really. So probably gone through that quite quickly, but just to summarize, we basically performed a large-scale survey, which sort of reveals measurable geometric variability within the PDB. So we can find that sort of we've got a lot of these outliers and clashes, and maybe they're sort of a bit more common than expected. We've used these sort of geographic style maps that we showed in the last slides that sort of aid in like visual diagnosis of outliers and help us to visualize, the extent of them. And the last point really, which is the purpose of this, is just to say that the underlying quality of this data should therefore be considered when we train algorithms. So we shouldn't just use, necessarily the entire protein data bank. There should be some sort of validation process. And validation processes do exist within the protein data bank. It's just, we've shown here really that there are still some examples that we might want to filter out and things. That's all the slides. If there's any questions.
Thank you, Will. Stop the recording and